Students, can I use Vietnamese to introduce you with our student and audience here? Yeah, thanks a lot. So I use Vietnamese. Sorry. Tiến sĩ Sudet Carol Matthew đến từ Ấn Độ. Thì tiến sĩ Sudet thì là nhà khoa học về y sinh, thì đã có 12 năm kinh nghiệm về nghiên cứu về cái ung thư, chẩn đoán phân tử, và cũng giống như là một số cái nghiên cứu ứng dụng. Ờ, Hiện tại thì tiến sĩ Sudet đang công tác tại SOPHiA Genetics. Ờ, ờ, anh chuyên về cái những cái giải pháp, cung cấp những giải pháp về genomics cho cái y học cá thể, cái precision medicine ở trong cái điều trị ung thư. Thì cụ thể thì tiến sĩ Sudet nó sẽ làm việc hiện tại làm việc ở trên cái giải trình tự DNA, và có giải trình tự cả RNA luôn, để tìm cái fusion, cái gen mà nó fill ở giống như nhiều vị trí khác nhau ở trên cái genome á. Và cũng phát triển những cái kỹ thuật sinh học phân tử, chủ yếu đây là kỹ thuật giải trình tự NGS thế hệ mới, để giúp cho các nhà lâm sàng có thể điều trị, cũng giống như chẩn đoán sớm bệnh ung thư ha. Thì hôm nay thì rất là vinh dự mời tiến sĩ Sudet đã đến với cái lớp của mình, đã trình bày những giải pháp ờ, của công ty SOPHiA cho cái ung thư đó. Cụ thể đây là mình làm về cái ung thư. Thì thật ra thì tôi định ban đầu á là mời ở bên chỗ hợp nhất, công ty khoa học hợp nhất, thì chủ yếu là cho cái platform giải trình tự của Element Bio đó. Thì cho cái máy AVT đó mọi người đó. Thì trong đây thì công ty hợp nhất thì có cái giải pháp về ung thư. Bởi vậy là cũng bên đây cũng là cơ hội để cho mình có thể được biết thêm những cái giải pháp mà nhiều công ty, đây cụ thể công ty SOPHiA Genetics đó cung cấp cho cái NGS về cái ung thư như thế nào ha. Mọi người có thể đặt câu hỏi thoải mái, để tôi có trao đổi với lại tiến sĩ Sudet á. Thì tiến sĩ Sudet rất là cởi mở và tạo điều kiện cho mình có thể hỏi, đặt câu hỏi thoải mái được không mọi người ha, bởi vì đừng có ngại. So thanks a lot for your spending time with us to give us the overview about the next gen of oncology. So please take on your. Thank you, Sudet.
Yep. Thank you so much for the wonderful introduction, even though I didn't understand the language, but I love to hear my name in a different language. Thank you for the introduction. So I'll give a quick intro, and I would like to switch off my camera for better connectivity. Okay. So am I audible to everyone?
Yes, yes, yes. I can hear you well.
Ah, perfect. So let me give a brief introduction about myself. I am Sudesh Karmathil, working as an implementation scientist at SOPHiA Genetics, based in India. It's been 2 years of long journey with SOPHiA, and it's a wonderful place to work. So today I'm gonna discuss about the topic like next gen oncology. Now, how SOPHiA DDM helps in get a high impact of the genomic analysis. So today's presentation focuses on SOPHiA DDM, a cloud native AI powered platform designed for high resolution genomic analysis in oncology. It integrates advanced secondary and tertiary analysis pipelines to accurately detect a wide spectrum of genomic alterations, including SNVs, indels, CNVs, fusions, MSI, and a lot more. So across the various cancer types. By leveraging the machine learning and proprietary noise reduction algorithms and a decentralized data sharing model, SOPHiA DDM enables scalable, reproducible, and clinically relevant insights from the complex NGS data sets. Yeah. So these are the few sections that we are going to discuss in today's topic. First one would be the SOPHiA genetics overview. And the products that we have from SOPHiA, the portfolio, like hereditary cancers, solid tumors, liquid biopsy, and the blood cancers. And I'll give you a brief introduction about the universal library prep, and also the how detecting the variants with the noise from that we get from the NGS workflow, and a brief about our SOPHiA DDM for genomics. So just give me a minute. Yeah. And the final slides would be about the conclusion that we have that we'll be discussing today in the today's presentation. So coming to the SOPHiA genetics overview. So, so this has been the possible thanks to our talented team of more than 500 employees spread across 28 countries. Our scientific impact is reflected by more than 300 peer-reviewed publications and by growing number of biopharma partnerships. The continued success of SOPHiA Genetics has brought us become a public company in 2021 with headquarters in Boston as well as Switzerland. And we have more than, as I said, we have like more than 500 employees, and out of which 30% of employees are like a PhD holders. So our mission is like to provide equal access to knowledge and capabilities by democratizing the data-driven medicine. We strive to provide equal access to the knowledge and the capabilities, since we strongly believe data-driven medicine draws insights from disparate complex databases to improve diagnosis, treatment, and drug development. And adaptable and scalable technology. So this is where we are at this point of time. Here you can see, we are currently exploring, these are the things that are presently available on SOPHiA DDM, the genomics for NGS. You can integrate your radiomics data, CT, MRI, PET scan, and also the clinical trials, along with your clinical and biological data as well. So we are now currently exploring the addition of proteomics and digital pathology data to match your current and future needs. So we have made our platform extremely agile thanks to our deep competencies in machine learning, pattern recognition, and predictive models, all of which can be applied to different data types and disorder areas. Okay, this slide gives you a update, what this means at this point of time. The data and the variants that are being called are across the globe, and which we do not know. Like let's just say if we have a variant which is pathogenic and that has been reported in Ireland, which someone from India, someone from Vietnam doesn't know that this has been classified as pathogenic. With the SOPHiA genetics community, we would like to connect all the dots across the globe. So whoever is using the SOPHiA community, when someone flags a variant as pathogenic, that can be seen on the SOPHiA DDM. So by this, it will reduce your consumable cost and customer dissatisfaction and churn and all those other things. Powered by AI capabilities, SOPHiA DDM analyzes and standardizes complex multimodal datasets to generate insights you need. The platform gives you access to one of the largest networks of connected healthcare institutions worldwide, where you can share knowledge, expertise with your peers to build a collective intelligence. We and concurrently breaks down data silos. The SOPHiA DDM cloud-based infrastructure is flexible and scalable, enabling to compute more data volume, support new applications, and functionalities according to your needs. Today, the platform has been already used to analyze more than 2 million genomics profiles through its 330 pipelines, and also across 750 more institutions, hospitals, and biopharma customers are using SOPHiA DDM for doing the analysis. This helps you to leverage AI to analyze and standardize the data, streamline your workflow, benefit from our network. A decentralized model. So what does it mean? So a platform operates to a decentralized model, so you can retain the custody of both sample as well as the data. So in the first step, you produce the data within your lab. Second, you'll upload them onto the DDM, onto the SOPHiA DDM for analysis. Third, you access the platform to interpret the results and gain insights from our growing community of experts for more informed decision making. So this slides tells you about accelerating the time from sample to the report. So the genomic data that we get from the NGS, so that is uploaded onto the DDM. Basically, we take the FASTQ files, which will do, upload onto the DDM, and we'll be doing a secondary analysis and tertiary analysis together on it. So which will take around 10 minutes for the tertiary analysis and nearly 4 hours for the interpretation, and like 1 hour like for the reporting. So all these things are like normally without SOPHiA genetics, but if you have SOPHiA genetic, all this can be done within an hour based on the expertise, of course. So addressing the burden of cancer and rare diseases. So in the slide, I'm just showing you the portfolio that we have. We have more than 423 applications that has for like hereditary cancers, solid tumors, hemonc, as well as the liquid biopsy, and 134 plus applications for the inherited and rare diseases. So here I'm going to explain you about the oncology knowledge in one place. The SOPHiA DDM platform has already dealt with many different genomic and clinical data early on. Our platform is constantly exposed to thousands of different data points coming from clinical data, variant classes, and different technologies, sequences, but also the enrichment kits. So we use the data to constantly fine-tune our algorithms and maximize our accuracy. Our oncology portfolio takes advantage of cumulative knowledge with hereditary cancers, also with over 50 solutions, solid tumors, and blood cancers. So for the solid tumors, with over 100 plus solutions, including DNA, RNA, and the ctDNA support. So here I wanted to discuss about the, the challenges that we face for different solutions, like, sorry, do you have any question? Okay. So here we are going to discuss the challenges that we face for the genetic variants across the hereditary cancers. So we have the main challenges are the challenging and laborious detect the CNVs, allo insertions, pseudogenes, and Boland inversions. And also the complicated workflows involving multiple providers, like let's just say if you're using a product with library prep from one person and doing an analysis on the other platform, which will be a little tedious to do. And the variable analytical performance, as well as the complex and the time consuming interpretations. So for this, we do have solutions from SOPHiA Genetics. So this is the overall workflow that we follow. So you have your sample, you'll prepare the library prep with capture. Once you prepare the libraries, I'll go with the sequencing on NGS machine. And once you generate the data, you upload the FASTQ files for the variant detection and annotation. And for the tertiary analysis, we use Alamut Visual Plus, which can be used for the visualization, interpretation, and curation. And also reporting can be done on SOPHiA DTM. So here, the total workflow takes up to 4 days. That includes your library prep to your report generation. So for the hereditary cancer solutions, we do have different options or different solutions. We have HCSV1, V2, clinical and whole exome solution, and also the enhanced exome where you can spike in our carrier screening, PGX, and hereditary cancer solutions. Apart from that, we do have some community panels for the community and custom panels for the hereditary cancers. And in this slide, we are going to talk about the solid tumor, like the challenges that we face when we do experiments for the solid tumors. So for the first thing is the tumor heterogeneity, and the second thing is the poor quality and small amount of starting material that we get from the FFPE samples. And we also have wide variety of complex mutations, and again, the demanding and time consuming interpretation doing it on different platforms. So for solid tumors, we have targeted applications like solid tumor solution and solid tumor for the DX like CEIVD, and also solid tumor plus, which is the RNA, which can detect the RNA fusions across like 143 fusions. And also we do have community solutions for the solid tumors. And in this, we are going to discuss about the hematology, like challenges of detecting the genetic variants associated with the blood cancers. Integration with the secondary methods is sometimes, need to be ensure a complete assessment. Like you need to do a MLPA, Sanger, or FISH, or different variants and fusions. Our solution offer a comprehensive with one NGS testing. We are able to detect the multiple SNV, Indel, and CNVs, and also the fusions. If the coverage uniformity is not good, you may have to cover you may have coverage gaps in some genes that need to be addressed with Sanger to avoid false negatives. So we are offering a great coverage uniformity on all the regions, uh, also for the tricky regions such as CALR, CEBPA, SEPPA genes, reducing the need for confirmation methods. So this is the brief portfolio that we have for the blood cancers. In this, you can see we have solutions for the myeloid, and also myeloid plus with extra genes, and also extended myeloid, which covers more than 98 genes for 98 genes, and the plus part for the RNA fusions can also be integrated to this. Apart from this, we do have two solutions for the lymphoid malignancies, the lymphoma and the community CLL clonality solution. Uh, Not only these dedicated solutions, we do have community panels for the blood cancers, and also the custom applications that can adapt and analyze genes relevant to your laboratory. So here, I'll tell you about the challenges that we are facing with the tumor or the liquid biopsy. Surgery or biopsy to obtain a tissue samples can be invasive, expensive, and time consuming, making it unsuitable for repeating the sample and longitudinal disease monitoring. In certain cancer types, it can be difficult to obtain sufficient tissue quantity and quality. For example, clinical studies show that up to like 20% of non-small cell lung cancers biopsies are inadequate for the molecular analysis. So the information provided by tissue biopsy is limited to a single point in space and time, falling to represent complex inter and intra-tumor heterogeneity. So these are the some of the solutions that we have for the MSK, that we have for the liquid biopsy. So I'm gonna tell, and the first product is MSK Access, which is our flagship product. Apart from that, we do have some community liquid biopsy solutions as well from SOPHiA Genetics. SOPHiA Genetics for universal library prep. So the library prep that we use for preparing the libraries from the SOPHiA solutions are it's universal. The why we have given this universal library prep. So the workflow for the wet lab workflow for preparing the libraries for both somatic and germline applications are same. So the only change that you see while you're doing the wet lab workflow is the probe change. So apart from that, the entire workflow remains the same. So I'll give you a brief about the library prep workflow in the next slide. So first, we'll go with the fragmentation and repair and A-tailing, which is a one step. And further to that, we'll ligate the adapters. We do a cleanup and amplify the product with the barcodes or the indexes that we add. And then we'll do the QC for the sample. Followed that, we'll do a capture workflow. All our solution users capture workflow. So what we do once you prepare the day one WGS library, we'll pool all those library into a single tube here, and then we'll be adding our blocking oligos and cod DNA, and we'll lyophilize the sample. So once the samples are lyophilized, we will proceed with the hybridization at 65 degrees by adding the specific probes according to the solution. So once we are like this hybridization can be done overnight or 4 hours depending upon your timings. So once we have our hybridized product, what we do, we pull down all the hybridized product that has been attached to the beads. Like we'll pull it to the beads. So you can see we'll be adding the streptavidin beads. Only the hybridized products will be bound to the streptavidin beads. And once this is done, we'll be further cleaning up the unbound DNA, which is not hybridized during the protocol. And then we'll further do the amplification, final amplification. And once the amplification and cleanup is done, your library is ready for sequencing. So this is the overall workflow that SOPHiA Genetics follows for the universal library prep. Now, the next section is like detecting the variant among the noise during the NGS workflow. So there are different types of noises that we get while we are doing a workflow for any of the solutions. There might be noise from the sample, noise from the sequences, noise from the sample heterogeneity, all these things. So I'll be telling you how SOPHiA DDM works, how SOPHiA DDM pipelines works to detect the variance, removing the noise from the NGS workflow. Yeah. So the numerous steps along the journey from sample collection to data analysis add various noises which can impact the detection of genetic variants from sequencing reads. Over the years, at SOPHiA Genetics, we have worked hard to understand these noises. So we have developed ways to decrease the noise at their source during data generation. All noises cannot be removed this way. We analyze data produced by various labs. We have consequently developed more powerful analytical tools that can accurately detect genetic variants despite noises, a feat made possible by our excellent understanding of noises. In this presentation, we'll present about the various noises that we get, and I'll in the next couple of slides. And you can see there are proprietary algorithms which can detect multiple things like SNVs and indels. I'll show you in the next slide. So analysis powered by SOPHiA DDM. So on the SOPHiA DDM, depending upon the solutions, you can call for gene fusions, SNVs, indels, CNVs, MSI, TMB, all those things. So these are our in-house algorithms, patented algorithms. We have given each of them a name, a spices name. So you can see the pepper, which is used to call the SNV and indel detection for the secondary analysis. Muscat for CNV and gene amplification calling, mustard for the MSI status, cardamom for the fusion detection, and mocha for our variant annotation on the tertiary analysis platform. So SOPHiA genetic application distinguishes the signal from noise to accurately and effectively detect variants from NGS data. So what we have done to do, to detect these variants confidently. So we carefully design our probes in such a way for all the genes across the sample, you have a uniform coverage. So our careful probe design ensures uniform coverage for high sensitivity and the precision. And the patented algorithms Our variant calling technology implements relevant analytical modules tailored to reduce linked to the sample type, sequencer, and the library prep. And also the CNV calling, our copy number variation calling modules adapt to experimental condition to perform double normalization to call CNVs missed by other tools with confident metrics. So for the CNV calling, I just would like to add one more point here. We do not add any baseline. So whenever you upload a batch of samples, we'll take the best sample out of that batch and use it as a reference and do a double normalization to call the CNVs. So in this section, I would like to explain you about how we design the probes and to optimize the coverage uniformity. So there are different methods to prepare the libraries. One is the capture based, which is enrichment, and again the amplicon based enrichment. So different targeted enrichment method leads to different types of reads and introduce different read bias. So if you can see, these are the probes, and this is capture based. On the right, you can see the amplicon based. So you can see there are like various gaps in between which are not amplified when you have done with the amplicon based. But when it comes to the capture based enrichment, you can see the probes are aligned for most of the region over here. So this is the one of the comparisons that we have done for one of our solutions. So especially for the myeloid here. So we have done a comparison for amplicon based as well as the uh, capture based from different solutions, competitor solutions, I would say. And here he can see these are the two genes. I mean, these are the genes that we are showing you here, which are really difficult to get the variants. So as you can see, the top tracks, 1, 2, 3, 4, all the 5 tracks that you see are from the amplicon base. So calling this TETS2, SEPPA, and FLT3 genes are really tough while using amplicon based. When it compares to the capture based, we have done it with 3 different users. The first one, which is marked in the red box, is from the SOPHiA myeloid solution. So you can see the TET2 gene is covered and TET2 and the other 2 genes are covered completely on the SOPHiA when we compared with the other solutions. So you can see there are gaps between the capture regions as well from the other solutions. So in this way, what we do, we design our probe designs are like so accurate so that we can call the variants for the difficult genes, which has like AT and GC rich regions, which are difficult to call. So again, like, I wanted to show you the coverage uniformity that we have for our solution. In this, you can see let's have a look at the SEPPA gene that is notoriously hard to sequence due to its high GC content and repetitive regions. Looking at the dotted 20% line, you can see the dotted line. Sorry. Most of the assays fail to it achieve it being well covered. However, I would like to point out one essay that actually has an increased coverage in the SEPPA. Well, to get such graph, the essay designer used a considerable amount of probes in this region, which means that they get a very high coverage, but the cost of through thousands of reads away in this regions. You can see it here. And this is the coverage uniformity that for the SEPPA gene from the myeloid solution from SOPHiA. The original coverage are different because these data come from different experiments with different sequencing depths. The value plotted here are relative coverages. The coverage of each region was divided by the median coverage. They are plotted on a log2 scale, which has been changed to fold changes to make it easier to read. So next topic is the distinguish the signal from the noise. So you can see there are different dots and different colored dots. So the first The red or the pink color shows the true positive, and we have blue color, which are false negatives here, and also false positive, and the black dots at the bottom are the false negatives. But there are three different dots, I mean, with a black outline, which is like, a difficult to call, because you can see the false positive as well as the true positive variant together in this solution. So this makes it a little tedious for us to get confidently call this variance. So the signal, the WAF input amount, the Poisson noise, and the enrichment bias and LCR are gives you the proper signal. When it comes to noise, when you have a lower input type, the input, the FFP input type, the FFP, the input amount, let's just say if you are using a lower amount, obviously it's really difficult to distinguish between the noise or the actual variant. And also the library conversion rate plays an important role in detecting these variants. Sample type influence noise levels. The, s- the sample origin will affect the quality of DNA and RNA that can be recovered. Samples that are immediately frozen provide the best material of genetic analysis. You can see the orange dots here, the orange bars towards the down. So these are the fresh frozen samples. However, many samples corresponding to the solid tumors taken during biopsies were fixed in formalin before being embedded in paraffin. So basically the FFPE sample. Such samples are suitable for producing images of tumor and the cells. However, the DNA or RNA in them get degraded during the storage. So the key message is FFPE samples show an excess C2T or G2A alteration, which are due to the deamination of the sample. Quantity input material drives the number of sequenced molecules. So what I'm going to explain in this particular slide is the input amount that you give while you start the library prep will show you the differences. So on the left side graph, you can see that we have taken a 2 nanogram, and on the right side, we have taken 20 nanograms. You can clearly see the higher input quantity of input material is associated with higher molecular diversity of sequence molecules, decreasing the chance of missing a variant over here. And a better understanding on the noise levels. So you can see the gold dots, which are true positives, and the blue dots, which are false positive. So depending upon the input material, the confidence level of calling a variant changes. Let's just say for the same sample, we have taken 2 nanograms to 50 nanograms. You can see there is a lot of noise that has been induced when we take a lower input materials. And thus you can see for the 50 nanograms, we can call the variants confidently as the input material is high. Just to add one more point here, sequencing more input molecules helps to decrease the false positive by reducing the noise that we get from the question. Okay, so in the slide, we have the sample, the potential origins of the noise. The first one is the sample. When we do an FFPE treatment, the deamination occurs during this. And during the library prep, we have the noises that we get from the DNA oxidation, fragmentation error, or the polymerase error. And the third noise that can be introduced in the samples are from the sequencing based errors. And also during in genome content, we do have homo and heteropolymers, high-low GC regions, homologous regions, as well as the long uh, tandem duplicate regions. With all these noises across the workflow, SOPHiA DDM SOPHiA Genetics wet lab protocol optimizes and helps you to call the variants on the SOPHiA DDM platform confidently by removing all these noises. So these are the background noise filters that we use for the Pepper technology. As you can see, for different samples, the noise levels are high for the tumor samples here. So when we introduce variant filters here, you can see the noise levels has been reduced. So in this, the R filtering based on the background noise removes the most potential false positives. SOPHiA DDM accurately detects the SNVs and indels. So in this, I'll be explaining you about the algorithm that we use to call the SNVs and indels, which is Pepper. So this is a sample with like without applying any filters, and also I'll show this is a deletion, large deletion that has been occurred in one of the sample, and we show how we realign this. So align, how important is the alignment? So this is a germline sample, but the same could be observed in somatic sample as well. In this case, we have both reads with deletions, black bar in the middle, and reads without deletion. So below those with the black bar where we can see the reads covering the deleted segment. So those colored regions is its regions are soft trim. You can see on the top track, these are the regions, and those are been trimmed, soft trimmed during the alignment, and it didn't reflect the deletion in the sample. The bottom plot shows you how we realign the reads to detect the deletion. So realignment enables effective indel deletions. Yeah. So this is with one of the samples how realignment works. The duplication is represented as an insertion when compared to the reference. So this is shown in a purple point in each read. So before realignment, the insertion point was inferred at different parts among the reads. The result is that the variance fraction of each insertion will be relatively low, and they might not be called with confidence. So after the realignment, the insertion points are located at the same position in all reads. The variant fraction will thus be high, so that the variant will be called with high confidence. So this is for the same insertion where we have done the realignment, and we can confidently call this particular deletion or the insertion. So this is another example of realignment enabling indel detection amongst the noise. So we have sample A represents 210 base pair deletion in gene MSH6, which is Lynch syndrome. Sample B, which doesn't have the deletion and is used as a negative control. Reads with the deletion needs to be anchored both sides. This has to be anchored both sides. This means the part of the reads to be need to be aligned with a point of reference. In this case, we can see that the right hand border of the deletion, that is the part where the reads need to be anchored, consists of a poly T which generates noise seen in the sample B, negative sample B. Detecting such deletions is challenging, but our tools rocket. So we have detected 200 base pair deletion in gene MSH6. Okay. The next topic is the SOPHiA DDM for the genomics. So what are the key benefits that we have? Here we represent the key benefits of SOPHiA, of new generation SOPHiA DDM with enhanced capabilities. The new platform will bring a unique workspace to host all modules, applications, and features with a central landing space to hub to facilitate the support and commercial technical information such as release notes. Everything will be available on it. So single platform to support both IVD and RUO applications, web access to simplify installation and uploads, and also this gives you a more analysis like revamped architecture, new capabilities, and the data protection. SOPHiA DDM for genomics, in this you can see the same information which I have shown in the earlier slides. So these are the things that we can call SNVs, indels, CNVs, translocations, fusions, genomic integrity, alternative splicing, and a lot more. These are our proprietary algorithms which can call all these variants. So SOPHiA DDM is a universal platform for detecting of variants associated with cancers and rare disorders. In-house algorithms and advanced technology accurately call, annotate, and pre-classify a wide range of variant types. This slide outlines the types of variants important for cancer and rare disorder analysis. At present, our proprietary in-house algorithms developed for secondary and the tertiary analysis of variants on SOPHiA DDM. So SOPHiA DDM ensures a safe flow of data for top-class data management and security. FASTQ files of raw genomic data are uploaded to the safe SOPHiA DDM, producing an output VCF file with the variant information. Following upload, data are encrypted and split, where sequencing data are separated from identifiers. A data service runs and moves raw data into an AI component where it is authenticated. The upload immediately starts variant identification and annotation. Data are decrypted when ready for the interpretation. The service segregates patient identifiers from the sample and stores them inside the client's domain with the Microsoft Azure cloud for integration with the final report. We do pseudo anonymize the raw data and analyze the results and store them securely. Services operate according to the regional data residency requirements. The system is secured by design to protect business assets and personal privacy information. So we are ISO certified and also IDVR and HIPAA and GDPR compliant company. So all your data that you upload onto the DDM are end-to-end encrypted for both RUO as well as the CE-IVD applications. So this slide gives you the information about the variant filtering that we do across the SOPHiA DDM. So for the data from the secondary analysis, SOPHiA DDM offers integrated filtering and prioritization features to narrow down the list of variants to a manageable number of relevant variants for further analysis. First, pathogenicity is assessed via dual pre-classification based on ACMG guidelines and machine learning. Interpretation can also be limited to a subset of genes using virtual panels. For example, using the OMIM or HPA browser. Custom filtering strategies with cascading filters allow quicker screening of relevant variants, and the familial variant analysis tool can be used to select inheritance mode and quickly identify the causative variants. So here you can apply different variant filters on the SOPHiA DDM so that you can easily narrow down your like your variant, like confidently narrow down your variants. SOPHiA DDM for Alamut Visual Plus OncoPortal work. I'll tell you what are the different softwares that are available from SOPHiA. So, and Alamut Visual Plus and OncoPortal work in harmony to help establish the clinical relevance of variants. SOPHiA DDM annotates variants and enable comprehensive variant filtering to isolate a manageable number of variants with potential clinical relevance for further evaluation. So Alamut Visual Plus provides additional comprehensive annotation of germline variants and enhanced visualization in a full genome browser. And the OncoPortal knowledge base enhances the interpretation of somatic variants by putting genomic biomarkers in the clinical context. So here you'll get the information about all your clinical trials, biomarkers, and all this information. And the third one is OncoPortal Mutational Tracker, which provides you a longitudinal view of the individual genetic markers over time to empower timely data-driven decisions. So in SOPHiA DDM for genomics, from data to insights, it can be done in five simple steps. So log in and upload your data, secure login with a two-step verification, quickly upload the FASTQ files, quick upload of FASTQ files, performance pipelines tailored to sample type, chemistry, enrichment kit, and the sequencer. And the second step is the variant calling and annotation, somatic and germline pipelines optimized to target exome-sized applications. Single or multi-sample trio analysis can be done. Advanced accuracy across multiple variant types in a single experiment. Detailed quality metrics. No bioinformatics expertise needed. Comprehensive annotation with up-to-date information from more than 55 world-renowned curated databases, guidelines, and predictors. And the third step here is the variant prioritization and filtering. Using the DDM is like user-friendly interface, variant pathogenicity levels assigned using machine learning complemented by guideline-driven ranking, multiple filtering functionalities to focus on disease-specific variants, create custom filtering strategies, or filter by inheritance mode. And the fourth step is exploration and interpretation. Integrated features are available to contextualize the variants, OncoPortal and the Alert Mutual Plus, which helps you, OncoPortal helps you for the molecular profiles are matched to the therapeutic, diagnostic, and prognostic clinical associations. Alamut Visual Plus for the rare and inherited diseases, variants are visualized in a comprehensive full genome browser. SOPHiA Genetics Community Insights further support variant exploration. And the final and the fifth step is like user-driven templates for customization, the report generation basically, CAP and CLIA compliant, user-friendly, guideline-driven information for decision making. So the reporting can be customized as per your templates. So this is one of the example report, I have just pasted here. So here you can see streamlined data are provided within one report. Germline and somatic customizable report templates ensure that all necessary information included to comply with the relevant guidelines. SOPHiA DDM produces concise but comprehensive reports, including critical information such as biological and actionable classification and therapeutic and clinical trial recommendations. So with this, I would like to conclude the presentation. So these are the steps that I would like to conclude for the presentation, which would with a better probe design, which helps you to save on costs and save time as you do not need to repeat the experiment. And with SOPHiA Genetics, we give a full onboarding support for adapting our solutions with the SOPHiA DDM Max Care program, cutting edge precision medicine, which will help you in evolving applications, and the end-to-end solution for multiple applications, which can be accessed on single platform for multiple tests. And also there's one more slide, so which I would like to conclude. Many factors affect the signal to noise ratio. SOPHiA genetic probes and the patented algorithms are designed to reduce noise and call variants with maximum sensitivity and precision. And the last point, SOPHiA DDM's pepper and muscat technologies accurately detect the SNVs, indels, and call CNVs with high resolution. With this, I end my presentation. Thank you. Now you can shoot your questions.
Yeah, thanks a lot, Sudesh. Very nice talk, very informative information about SOPHiA service and software.